started. So it's my distinct pleasure to introduce Qin Yan to us today. He's one of the invited speakers for this year for the melanoma program. So for those that don't know, melanoma program is a fairly well-established one going back to the 1980s when we started the first, well, it wasn't me, other folks started the first interdisciplinary disease team. John Kirkwood and Steve Arians specifically. And then as the years went by, Ruth Halliban, who's sitting here, wrote the first version of the Yale spore in skin cancer, first funded in, I think, 2006 or 7, Ruth, something like that. We just submitted the 4th iteration. So one of the best things about working here is actually our colleagues, and I think Qin actually exemplifies that. So he came to us from Harvard, where he worked in the lab of Bill Kalin, actually on epigenetics and renal cell carcinoma. But at some point, it became clear that some of the things that he was studying were very applicable to melanoma as well. And he submitted a developmental research project to the spore in its previous iteration, and that subsequently blossomed to a full project. We are thrilled to have Qin working with us. We couldn't ask for a better collaborator, both in terms of his scientific depth and in terms of his personality. He's definitely one of us, and I actually don't care that he's the scientific co-director of the breast unit. As far as we're concerned, he's ours. So without further ado, Qin, the floor is yours. Thank you.
Well, thank you, Harry, for your kind introduction. And I would also like to thank the melanoma program for nominating me to present here. I would say Cancer Center Ground is one of the events that actually led me to work on melanoma. And on my way back from a ground rounds talks, and I was working together with Marcus Bosenberg about a decade ago, and we were talking about GERY1B, which might be important in melanoma. I was working on GERY1B because I generate knockout mice. And well, we just started the collaboration, and it's a very fun collaboration. And this is something I'm going to tell you today. So let me get this started. Fix the pointer. So this is my disclosure. So what I'm going to do is first to give you a very quick overview of cancer epigenetics, and then you'll tell you 2 stories related to this histone demethylase KDM5B, how it regulates drug resistance and immune evasion. So as many of you know, the epigenetics is study of hierarchical traits that does not depend on the underlying DNA sequences. And the major epigenetic mechanisms include DNA methylation, nucleosome structure, histone modifications, and non-coding RNAs. There's a number of regulators of epigenetic mechanisms, including the chromatin remodelers, which are involved in moving the nucleosomes around, and writer, eraser, and the readers of histone or DNA modifications. So what I'm going to tell you today mainly focus on KDM5B, which is an eraser, which is involved in removing a certain modification, and the ZDB1, which I'm touching upon, and which is a writer protein. So many of you are quite familiar with these hallmarks of cancer. And what I'm going to tell you a little bit about is the immune evasion that cancer cells have to achieve. And if you look at it on the right side, this is those are new hallmarks that have been added to the hallmarks of cancer, and 2 of which actually quite related to epigenetics, including unlocking phenotypic plasticity and epigenetic reprogramming. So that's what I'm going to tell you today. So as many of you know, epigenetics can regulate many of the cell fate, and also a lot of mechanisms are involved in anti-tumor immunity. And just on the tumor cells, for example, it has been shown DNA methylation and histone modifications have been involved in regulating tumor antigen expression, cytokine secretion, PD-L1 expression, and also chromatin structure has been shown to be important to respond to cytotoxic attack. And those modifications are also important on other immune cells, including cytotoxic T cells, dendritic cells, and the macrophages, which is not depicted here. So just a brief introduction on my laboratory, and we are interested in cancer epigenetics, of course, and one of the areas we are interested in is cancer metastasis. Shown here just one of the example where we showed one of the target called CECR2 is a driver of breast cancer metastasis. And you can look at here, if you knock down CECR2, you can suppress the ability of those breast cancer cells to metastasize to the lung. And if you overexpress CECR2, you can rescue this phenotype. And of course, we are very interested in immune evasion. Part of the talk I'm going to talk about then, and this is something that I will mention later. And so I'm not going to go over this figure. And we are also interested in drug resistance. And I will tell you about our work on drug resistance in melanoma. But this is a diagram actually from a breast cancer work where we show that trastuzumab resistant cells have increased oxidative phosphorylation. And if you block oxidative phosphorylation with oligomycin A, if you combine that with trastuzumab, you can actually regress the tumor formation by the drug resistant cells. As a matter of, because we are interested in those areas, we are also interested in developing epigenetic drugs. And I will tell you some of our work on KDM5 inhibitor development. And this is some work that we have done a couple of years ago, where we characterize a potent bromodomain inhibitor, where we show that this bromodomain inhibitor, NHWD870, can not only inhibit the ability of the tumor cells to grow, but you can also hit on the macrophages by suppressing the expression of CSF1, a critical regulator of macrophage polarization and the macrophage proliferation. And this drug actually have entered the phase 1 clinical trial in China and moving into phase 2 very soon. So my laboratory has been focusing on a group of enzymes called KDM5, histone demethanases. And as you can see here, this group of enzymes have 4 of them. They are called KDM5A, B, C, D, or GERD 1A, 1B, 1C, and 1D. And all of those have this JMJ-C domain, which is the Jumanji C domain. It's a hydroxylase domain. And by hydrogenation of the methylation group and the removal of formaldehyde, they actually can demethylate the histones. So this group of enzymes can demethylate tri and dimethylate lysine 4 on histone H3. And because those methylation marks are critical marks for actually transcribed genes, so by doing so, this group of enzymes can silence transcription. But that's not the whole story. And those proteins actually have other domains, including AT-rich interactive domain, which is involved in DNA binding, and some of the PhD fingers, which are involved in binding specific histone modifications. In addition, they can interact with many other proteins involved in chromatin remodeling and chromatin transcription regulation. So they have, it has been documented that this group of enzymes can not only be transcription repressor, they can be transcription activator in some other settings. So today's talk I will be focusing on this protein called KDM5B or GERD1B. Also another known name is called the PLU1, because there's a number of evidence showing that KDM5B has oncogenic role. It was shown to be overexpressed in many cancer types, including skin cancer. Initially, it was identified as a downstream gene downstream of HER2 in breast cancer, because it was shown to be downregulated by anti-HER2 antibody in HER2 overexpression cells. And it has been shown by Nani-Polyan's group that it's amplified in luminal breast cancer, and it's a potential luminal lineage driving oncogene. And we have shown in mouse memory epithelial cells that GERD1B can recruit GATA3 to regulate FOXA1 expression, and that contribute to estrogen receptor target gene expression. And in fact, if you look at the estrogen receptor positive tumors for breast cancer, higher activity of GERD1B or KDM5B is correlated with poor prognosis of those patients. And Nanny Polyak group has also shown that KDM5B can promote transcriptomic heterogeneity, and this actually contributes to the therapeutic resistance. And this is just one of the mechanisms that this could contribute to resistance. I will tell you more about our work on a different angle. In addition, when we deplete KDM5B, first initially in breast cancer cells in syngeneic mouse model, you can see downregulation of KDM5B can decrease the ability of those tumor cells to grow. And it was shown by Minha and Henin's group that if you suppress KDM5B expression in melanoma cells, initially those tumor cells actually grow faster. However, after serial transplantation, those cells still crash, so suggesting that it's required for melanoma maintenance instead of proliferation, initial proliferation. And it was shown in multiple groups, including ours, that KDM5B is involved in drug resistance. And shown here just one of the example by a company, Constellation, where they showed in multiple cancer cell lines, including melanoma here, if you compare the effect of KDM5 inhibitor on parental cells or drug-tolerant persister cells. If you actually, in this case, they did a pre-treatment of those cells, and they have shown that the KDM5 inhibitor cannot inhibit the growth of the parental cells, but they can prevent the emergence of the drug-resistant tolerant with DPT cells or drug-tolerant persister cells or drug-resistant cells. In prostate cancer, if we cross the KDM5B knockout model to the P10 knockout model in prostate specific deletion, and where you can see P10 knockout model can form prostate cancer. But if you get rid of KDM5B, you can normalize those prostate tumors. Basically, you can see the size is much smaller. Now, I want to move back to melanoma because this is a focus on our talk today. First, when we looked at the TCGA data set, this was done by Goran. I attended the graduate student at that time in Marcus Bosenberg, who is a fellow right now, where he showed that high KDM5B expression is associated with poor survival of melanoma patients. So now we decided to look at the melanoma when we followed some of the work from Marcus Bosenberg's lab about the melanoma propagating cells. It was published more than a decade ago that if you look at the mouse melanoma cells, you can sort them to three different populations: the P75 positive cells, CD34 positive cells, or the double negative cells. If you look at the ability of the cells to form tumors, the CD34 positive cells can form tumors very efficiently, and the double negative cells can do so with less efficacy, but still works. And the P75 positive cells do not actually form tumors if they put them into mood mice. So we decided to look at this more systematically, and when this is just a diagram, a table showing many of the Yale University mouse melanoma cell lines generated by Marcus Bosenberg's laboratory. And those cell lines are generated from BAC6 animals. And you can use those cells for syngeneic transplantation experiments. And the two of the cell lines we used here are YAM 1.7, which I will use it also later on for immune evasion studies, and also YAM 3.3 cells. The reason why we chose those cells because they only have two populations: CD34 positive and CD34 negative. Both of them can form tumors. So this provides a nice system to look at the population changes when we put drugs onto them. So we used because those are BRAF mutant tumors, and we treat those cells with the BRAF inhibitor. In this case, we actually use the PLX4032 or verorafenib. As you can see here, if you compare the parental cells, and you have more CD34 positive cells, if you look at the drug resistant cells, you have, which we designate as YUM, YAM 1.7 RE-R or resistance. They have more CD34 negative cells. When you look at the effect of the BRAF inhibitor on those subpopulations, you can see CD34 negative cells are more resistant to BRAF inhibitor treatment because there's less growth inhibition. And this phenomenon is also reversible. If we treat those, you can see that they shifted to the left side, meaning CD34 negative cells. However, if you remove the drug after a couple of passages, and they were shifted back to the parental cell population. So one of the things that was Xiaoni, who was a graduate student when Marcus lab, basically noticed that there's an increased expression of KDM5B if we treat those cells with BRAF inhibitor. And this is shown in YAM 1.7 cells, 3.37 cells, but also when you compare the parental with the resistant cells, you see the similar increase of KDM5B expression. And this is reversible. If you take out all the BRAF inhibitor, the KDM5B expression drops down, and it's shown in YAM 1.7 cells as well as 3.3 cells. So when we did the genetic experiment, when we knocked down KDM5B expression by shRNA, as shown here, we can see in the YAM 1.7 cells, there's a decrease of CD34 negative cells after we deplete KDM5B. When we look at the phenotype, and it's consistent to what other people have seen in other melanoma setting, if you knock down KDM5B, you actually increase the ability of them to grow in vitro. And then those cells are actually more sensitive to BRAF inhibitor treatment. So this is not only true in mouse cells, but also in human cells. This is UMAX cells. If you knock down KDM5B, you can see induction H3K4 trimethylation, which is the substrate of this enzyme. And you can see those cells grow faster. However, they are less sensitive. They are more sensitive to BRAF inhibitor treatment. And if you look at this in animal models, similar things happens when we treat cells with BRAF inhibitor, and you can see KDM5B level increase. And if you take away the BRAF inhibitor, you can see the KDM5B level drops down. So, and then we look at if you look at the population of those cells, you can see increased CD34 negative cells when we treat the cells with BRAF inhibitor. When you take out the BRAF inhibitor away, and then those was normalized. So this is all consistent with our data and others have shown, which are not shown here, that the KDM5 inhibitor can suppress the emergence of drug-resistant cells. So to summarize this part, basically we have shown that CD34 negative cells are more resistant to BRAF inhibitor treatment, and the BRAF inhibitor can increase CD34 negative cells, and it can induce KDM5B upregulation, and this is reversible. And KDM5B loss can reduce this population cells and induce drug resistance sensitivity. So now I want to switch gear to talk about immune evasion. And firstly, I want to start with this cancer immunity cycle, which many of you have seen before. Basically, this is a diagram showing that the cancer cells interact with the immune system. and there are many ways that cancer cells have adopted to evade immune evasion, to evade the immune response. So as a matter of fact, because of this mechanism, and some drugs has been developed, including the anti-PD-1, PD-L1, anti-CTLA-4 antibodies, as well as the ways to boost the effect on the T cells. However, there's not much is really actually known about the trafficking of T cells to tumors and the infiltration of the T cells into the tumor at that time when we started. And what's known about epigenetics in this setting, many of you are quite familiar with this concept about cold tumor and hot tumor. The cold tumor are not really responsive to a lot of treatment, but the hot tumor will enable them to be responsive to immune checkpoint blockade. And a subcouple of epigenetic regulators have been shown to be critical for this cold to hot transition. And if we treat this the tumors with a couple of inhibitors against those targets, like DMNT inhibitors, OSD1 inhibitor, EZH2 inhibitors, you can shift them to be more hot tumor, become more hot. In some of the settings, not in all the settings. This it's kind of related to what we are trying to do. And at that time, actually a couple of years ago, before that, we have looked at the KDN5B and how it's related to other genes when we look at the TCGA melanoma data set. And to our surprise, actually, KDN5B expression was shown to be negatively correlated with many of the immune-related genes. And if you look at those top signaling pathway, those are all immune system-related genes. Those are negatively correlated with KDN5B expression. If you look at the identity of those genes, those shown here are the gene names, and on the right side are the Spearman score. And you can see many of the cytokines, for example, interferon gamma and TNF-alpha, CXL9, CXL10, which are involved in T cell recruitment, are all negatively correlated with KDN5 expression. And some of the targets for immune checkpoint blockade, PD-L1, CTLA-4, also negatively correlated with KDN5 expression. This will be important when we are trying to look at the immune checkpoint blockade resistant tumors. So when we look at the KDN5B expression, protein expression in melanomas, and if you compare non-responders and responders, we can see increased expression of KDN5B, which is shown in red, in those non-responders compared to the responders, and the quantification is shown here. So this motivated us to look at the role of KDN5B using animal models. So as I mentioned earlier, Marcus Bosenberg's lab had generated a series of YUMM or YUM models. One of the models that we started to use is this YAMA 1.7 models. This stands for ER, stands for exposed to radiation, meaning those cells will radiate so that they have more mutations, they can generate more antigens that can be recognized by the immune system. So when we knocked out KDN5B in those cells, as you can see here, those cells initially can grow. Then they got fully rejected after a while. And then more importantly, when we challenge those animals with the cells, control cells, which normally can grow very well, they never grow up. So this is very important. Imagine that if you have treated a patient with a drug, and for example, in this case, KDN5 targeting drug, and those patients will not have recurrence because they should never grow up because of immune memory response. So this actually translated to 100% survival, which is great. And then we looked at the T cell infiltration when we compare the control cells and KDN5B knockout tumors at the very early stage. You can see the T cell infiltration either by immunohistochemistry as well as FACS analysis. And there's another way to say that this is immune system dependent. We compared the ability of the cells to grow in wild type mice or RAG deficient mice. And as you can see here, the B6 is the wild type. Those two curves are what I have showed you before. And if you look at the ability of the cells to grow in RAG deficient mice, the control goes here, and then KDM5B deficient ones grow kind of similarly, although slightly slower. So this basically set up the stage that KDM5B is critical for immune evasion. So the next question is, what's the mechanism, right? So how to understand this, we did RNA sequencing comparing YAMA 1.7 cells KDM5B knockout versus wild type. And we can see there's an induction of a lot of signaling pathway involved in DNA RNA sensing pathway. And showing here the gene set enrichment analysis, the enrichment plots, where you can see there's an enrichment regard like receptor pathway, cytosolic DNA sensing pathway. Those are all induced after you get rid of KDM5B. So now, how does this actually work? And are those sensing pathways critical for the function of KDM5B? As many of you know that the double-strand DNA and double-strand RNA are sensed through those pathways, and double-strand DNA is sensed through cGAS-STIN pathway, like TBK1, F3 or F7, and the interferon response, and this leads to the induction of interferon stimulated genes. The double-strand RNA could be sensed through RIGA MDA5, MAVS, or TOR3, and basically also signals through and activate interferon stimulated genes. So what we did is we knock out each single component through this pathway and see what happens. When we knock out the KDM5B, as you see, it does not grow, and the wild type cells do grow. If we combine that with knockout of the MAVS or STIN and the important mediator of double-strand RNA or double-strand DNA sensing pathway, you can see partial rescue right here. If you get rid of both of them, you see much better rescue. So we went on and went upstream. When we get rid of the cGAS or MDA5, you can also see partial rescue. If you get rid of both of them, you can see pretty good rescue response in this case when in two independent clones. So having established that, now we want to understand why those pathways are activated. So one of the things that we noticed is that when we compare the control cells with the KDM5B knockout cells, we can see the induction of double-strand RNA in KDM5B knockout cells. And we have seen this also in tumors as well. And this motivated us to go back and to reanalyze our RNA sequencing data for expression of those retroelements. And those retroelements are part of junk genome, and that people are normally ignore. And it turned out to be very important in this case. And what we have seen is that if we knock out KDM5B, we can see induction of those retroelements, and especially some of the endogenous retroviral elements. And the one of which is called MOV30, and you can see multiple of those showing up. And MOV30 is actually critical for the interferon response, because if we knock down MOV30 with siRNA, as you can see here, you can see the downregulation of interferon stimulated genes, suggesting that this is at least partially contribute to the interferon induction and maybe the response to immune evasion. And one thing that we were puzzled about is that since I showed you that both DNA and RNA sensing pathways are required, where are those DNA coming from? And we postulated that those DNA will be coming from reverse transcription of those RNA species that would generate through after we get rid of KDM5B. And one experiment we did is use reverse transcriptase inhibitor. This is a cocktail of reverse transcriptase inhibitor that's used for HIV treatment. And where we see if you treat the cells with those reverse transcriptase inhibitor, you can see suppression of the interferon stimulate gene expression, suggesting that this DNA might be created through this pathway. So now with all those mechanisms, now the question is, can we translate it to targeting this? The quick question is that, can we induce anti-tumor immune response with KDM5 inhibitors? So as I mentioned, because there's a lot of evidence showing that KDM5 are critical for cancer initiation, progression. So we have started working on this by multiple methods to develop KDM5 inhibitors. So initially, with a pilot grant from Yale Small Molecule Screening Center, now called Yale Center for Molecular Discovery, we have done some screening, biochemical screen for KDM5 demethylase inhibitor. And initially we did 100,000 compounds. With those as a preliminary data, we were able to obtain support for an NCI experimental therapeutics program, where we were able to assemble a team about 30 scientists to develop those inhibitors. So we have done a high throughput screening, about 200,000 compounds. Those are high quantity compounds, and have done extensive medicinal chemistry optimization of some of the compounds. And we have solved 35 crystal structures, KDM5A, with different inhibitors. And shown here just two of them, basically showing that they combine very tightly to the active site. One thing that I want to mention, that those inhibitors are all pan-KDM5 inhibitors. They hit both all KDM5 family members, because the catalytic site is very similar for all those KDM5A family members. So even with those, and we decided to ask what the KDM5 inhibitor can do. And the one thing that we decided to do is we selected 4 KDM5 inhibitor here. Those are high quality specific KDM5 inhibitor. As you can see here, they all induce H3K4 trimethylation, which is the substrate of the reaction, then did not do anything to the other of the histone modifications. And we did those actually in MCF7 and multiple human breast cancer cells. And when we looked at the gene expression changes, to our surprise, we see the top pathway that's upregulated are those interferon signaling pathway. At that time, I was like, interferon pathway is not something I want to work on. Not so much now. So anyway, so where we see is that there's an induction interferon pathway, and we have seen this in multiple cell lines, multiple drugs, and we were able to understand how this actually worked. And at the end, we were able to show that the KDM5 inhibitor can induce H3K4 trimethylation at the STING promoter, and by doing so, it actually induced STING expression. And this leads to the interferon stimulate gene expression, and this leads to the T cell infiltration. So this is a little bit different from what other people have been trying to activate this pathway through using STING agonist, which the limitation of those drugs is in many of the cancer cells, you actually have STING silence. So by inducing STING, and this provide another mechanism how we can activate this signaling pathway. So now, and we actually tested KDM5 inhibitor in multiple human melanoma cells, and we can see induction of STING, and in this case in Western blot here, and induction of interferon stimulated genes. And so we thought this is a sure bet, and the KDM5 inhibitor is going to work. And to our surprise, nothing happened. And when we put this in the mouse melanoma cells, the YAMA 1.7 cells, the model system that we have tested, two KDM5 inhibitor, and the retro element were not induced. The interferon stimulated genes were not induced. Nothing happened. So we did not want to give up because we thought maybe there's some limitation of those drugs. And so we did those rescue experiment to understand whether the kinetic activity is required or not. So what we did is that for KDM5B knockout YAML cells, we reintroduced either wild type or mutant KDM5B into those cells. Those mutant are catalytic dead KDM5B. And as you can see here, in both cases, you can see wild type or mutant KDM5B can suppress expression of the retro elements and those interference stimulating genes. Moreover, both of those can induce the growth of those tumors. So now what? Now we are back to the starting point and kind of depressed right at the time. So we went on and decided to look at all the repressive mechanisms and to see which one might work. And one of the things that we decided to look at is EZH2 inhibitor, for example. And those are two higher quantity EZH2 inhibitor. It did not do much either. And then there's some clue that H3K9 methyltransferase would work, and we use a pretty dirty, actually, H3K9 methyltransferase inhibitor called chetosine. And it can inhibit H3K9 trimethylation. You can see induction of MOV30 and some of the interference stimulating genes. So now there are multiple H3K9 methyltransferase, and so we knock out each single one of them to see which one is critical. When we knock out G9A or SU39H1, it did not really do anything. But when we knock out ZDB1, which is shown here, you can see robust induction of MOV30. So this was great news. So at that time, we were quite excited. And then when we did co-immunoprecipitation experiment, we actually can see that KDM5B can interact with ZDB1 when we did ZDB1 IP. There's a pull down of KDM5B by ZDB1. And then this is quite exciting. Then we decided to map the binding of KDM5B and ZDB1. And shown here just the heat map where we ranked those KDM5B target genes, where you can see KDM5B combined then very well in wild type cells, not so much in knockout cells. When we look at ZDB1 binding, you can see amazingly overlapping binding of ZDB1 and H3K9 trimethylation, which is the product of ZDB1. H3K9 trimethylation is a repressive mark that can suppress gene expression. And to our surprise, when we look at H3K4 trimethylation and dimethylation, which are the substrate of the KDM5B, you can actually do not see much effect, suggesting that KDM5B function, demethylase function, is probably silenced in this setting. So now those are all important, and now we want to look at this in a drug resistant setting. And in this case, immune checkpoint blockade resistant setting. When you look at the KDM5B expression, it's actually lower in the patient with complete response to anti-PD-1 blockade compared to the ones with progressive disease. So this suggesting that if we can lower KDM5 expression, you can make the resistant tumor sensitive. Indeed, that's actually true. And we use this YAM 1.7 model, which is the parental model for the YAML 1.7 I have showed you before. This model is resistant to all immune checkpoint blockade, PD-1 blockade. If you look at this, nothing happens. If you throw CTLA-4 antibody on them, nothing happens. If you combine them, still nothing happens. In this very refractory model, you can see if you get relocating KDM5B, you can already see some response. If you combine with PD-1 blockade, you see synergistic response. It can extend the survival of those animals. You can basically double the survival of those animals. And this is just one of the PD-1 resistant model. And when we look at another model, which is the YAMA interferon gamma resistant model, you can see similar phenotype. So lastly, is this also true in humans? When we compare the KDM5B expression with the endogenous retro elements, part of the category of the retroanimals. You can see the ones with higher KDM5B expression was shown on this. You have lower expression of some of the ERVs shown here, just one example, ERV 2637, and it's anti-correlated with KDM5 expression, and the expression is correlated with a better response to PD-1 blockade. It's opposite to what we see with KDM5B. So basically to summarize this part of my talk, which I showed you that KDM5B can interact with ZDB1 and it can recruit ZDB1 to the targets to deposit H3K9 trimethylation to silence retroanimals. If you get rid of KDM5B, you can activate endogenous retroanimals, you can activate double-strand RNA sensing pathway and double-strand DNA sensing pathway through the reverse transcription process. It leads to the better representation of the MHC1, and the cytokine secretion lead to higher immunogenicity and a better response to immune checkpoint blockade. So although we're the first group that show that KDM5B is critical for immune evasion, we are not the first group to show ZDB1 has this function, and the multiple groups at about the similar time show that ZDB1 is involved in suppressing tumor immunogenicity, and this is just multiple papers basically by multiple groups. And this basically add to the what do we know about epigenetic regulation of the viral mimicry pathway. Basically, I have showed before that DNMT and LSD1 can do this, and here we just show that KDM5B and ZDB1 can do this. And all those inhibitors will be able to induce the viral mimicry response, interferon response, and the better response to immune checkpoint blockade. So now I would like to thank all the people involved in this, especially Marcus Bosenberg's group, and where we had the fun collaboration, a decade-long collaboration. And the drug resistance work is led by Xiaoni Niu and Samin Zhang. And the immune evasion related work is led by Samin Zhang. Samin has actually studied her assistant professorship at Shanghai Tech University. And some of the bioinformatic works are done by Wesley Tsai and Goring. And I also like to thank all the Yale SPOR members for their kind help through the course of this project. And when I tried to start on melanoma, the SPOR members welcomed me with open arms, and that's how I can get where we are here. And I also like to thank all the other funding agencies for their support. As you can see, a couple of melanoma research foundation, Melanoma Research Alliance have been very helpful in supporting our research in melanoma. And I would like to thank you all for your attention, and I'll welcome any questions. Or maybe one back. Yeah, that's a great. So the question is whether we have tried to combine KDM5 inhibitor with a STING agonist. That's a great suggestion, and we have thought about this, but we have not had the time to do this experiment. Yeah, which we should have done. Yeah. Okay, Harry.
That was a great talk. Thank you so much. Went back and forth a little bit between KDM5 inhibition and KDM5B specific inhibition, and I know that in melanoma you think that KDM5B is the most important one. What about other tumor types?
We have actually been working on breast cancer and also some other cancer types. Where we have seen is that actually maybe I'll show you one slide here. This was just published. Basically, this is MC38 with the colorectal cancer, where when we treat those animals, tumor-bearing animals with the KDM5 inhibitor, you can suppress the ability to grow. So it works in colorectal cancer. Also, when we look at the breast cancer, you can see they have some efficacy. You can also combine that with PD-1 blockade. We can have, I would say, additive effect. So it works in multiple cancer types. It's just where we need to find the correct cancer types and subtype even to so that we're able to use those inhibitors.
And these are all pan-KDM5.
Yeah. Yeah, so yeah, those are all pan-KDM5 inhibitors. So one of the things that we are trying to do is to develop KDM5 family member specific degraders, and with the because with the PROTAC or some other similar kind of mechanism or molecular glue type of mechanism, you can develop a specific degraders against KDM5, and we are actively working on that. We have some potential degraders that work specifically on KDM5B, and some of them work on multiple or KDM5A in different settings.
There's an online question from City Chen. You can't see it. I'm happy to read it.
Yeah, I can read it. So the question is, do I anticipate other epigenetic reader and the writer to have similar effect? Yes, because actually I have showed you one of the diagram. There are multiple other ones on this. Okay, here. Ones which have been shown have similar effect, and although I have to say in different cancer types, and they have different effect, and we just need to find the right one that work in our setting. Okay, that's.
I got another question for you.
Okay.
So you showed that KDM5B was anti-correlated with all sorts of immune factors, both positive and negative, including FOXP3 and immune suppressive. What about the cell types? Did you look at correlations with presence of NDCs in the tumors, those sorts of things?
Oh, that's a great question. We have not looked. Yeah, so just need to do some single cell analysis to, or just analyze single cell data to see that. Yeah, it's great suggestion. Yeah, should do that. Arnold.
Wonderful mechanistic talk, and I love how you correlate that with relevant data. My question is regarding KDM5B and its effect that seemed to be enzymatic independent. That you showed us. I was wondering whether they would be inhibitors that could inhibit the scaffolding effect of KDM5B and its interaction with ZDB1, and whether those could be more appropriate for look at PD-L1 increased response.
Yeah, that so Arnold, the question is whether we should inhibit the scaffold function of KDM5B, which is great. So that's something that we are thinking on the way, because we have to, first of all, we need to identify the domains that are critical for those interactions. And then one of the things that we're trying to do is to look for those domains that are involved in interacting with ZDB1, and then those inhibitors will have more specificity, as you suggested, to target this pathway. And it's probably better than KDM5 inhibitor or ZDB1 inhibitor, which might have some other off-target effect that we don't want to see.
That's quite interesting, because when you think of, for example, LSD1, there are inhibitors that seem to be targeting the enzymatic domain of the demethylase, but we know that they actually impact scaffolding effects in creating broader proteins that play a role in LSD1 mediated tumorigenesis. So I wonder if those types of inhibitors are out there.
Yeah, it could be made, but we don't have those yet. Work in progress. Okay, if no more questions, thank you.